So what I'm trying to do is switch gears a little. Bob explained how DNA barcoding works and how it's applied in food security and in other applications, but I'm switching gears in a matter that I'm looking at biodiversity assessments. And I'd also like to present you a few ways how we integrate not only our scientific colleagues and friends all over the place, but also citizen scientists, families, schools, for that matter. And it revolves around one topic, and that's what I call biodiversity assessments. Very often you'll find the term environmental assessments. So when you look at all these images that I have here on this slide, most of them involve the process of an environmental assessment, which is basically going out in a given region that is probably planned for, or people want to log in there. It's new ground for agriculture, or designed or dedicated for urban development, or even mining, so any kind of resource extraction. But before people just go there and do whatever they're about to do, they have to go through a very rigorous assessment process. That means people go there, professionals, and determine what lives there. And in addition to what lives there and how many of those, they determine how unique is this biodiversity in that given region. Is it only occurring there, which would probably prevent one of those things going to happen, because if the neighborhood has not the capacity to just bring in the or cover that impact humans have on their environment, then there's a problem with that. And we also do assessments to do what you see in the first image up there for building reserves for protected areas. If you want to safeguard and keep animals and plants in a certain area, we just really have to figure out who lives there and why is there a value in protecting it. Well, how have we been doing that in the past, or we're still doing it that way for the most part? Well, we focus on what we call indicator species. So there's a little school, elementary school level definition what an indicator species is. It describes the characteristics of a given environment. So you can pick certain species, few of them, that are characteristic for a certain environment. If you find them, that's probably a good sign. If you find other species that indicate a bad influence, then this is a bad sign. So some of them probably jump on very disturbed environments where humans had already an impact. The number of these indicator species in total is relatively low, and that's due to the fact that people that work in that business have to give their answers quick. And quite honestly, if you have to identify every living thing in a given environment, you pretty much all scientists will be soon at an end, and they haven't defined everything because it's so difficult. Morphological identification, especially if it's nothing of that, but smaller. Imagine flies, tons of flies. They all look alike to you, but probably there's 400 species of them hiding in a little pile of them. So the question is, how can we do a really thorough assessment of an environment without having to fall back on these kinds of methods? Well, DNA barcoding might be the answer for that, because with modern DNA sequencing technologies, you can actually sequence pretty much everything you have in a given sample. And I want to give you an impression what it means when I talk about sample, and I'd like to explain you how we get these samples. So you're looking at something a colleague in Britain calls insect soup. So these are jars full of insects. So the dark stuff there is not any kind of dirt. That's actually really just individual insects. So some of these jars can contain thousands, literally thousands of individual specimens, and probably hundreds and hundreds of species. The old-fashioned way, and we do it partially, would be dump that on a petri dish in front of you and start sorting. Sorting them individually, probably first in piles of things that belong together, like all flies, all wasps and bees and all that. And that's as far as we go in the lab at some point. But then you would go there and try to identify everything. You have a lot of books and try to compare that. So that's a pretty tough job to do. How do we get these insect soups? Well, that brings me to a very interesting device. It's called a malaise trap. It has nothing to do with the French word malaise in that sense. It has nothing with sickness or disease. René Malaise was a Swedish fly expert who developed this kind of method in the 1930s in order to catch flying insects. So the way this thing works is you put that somewhere where you know insects are flying around, and it's what we call an intercept trap. So it's in the way of their path. They have to get around it. Unfortunately, if you look at this fly here, they'll enter that, and there's only one way for them out of it, and that's this bottle on top that's filled with ethanol. The thing is that the middle mesh stops them from flying, and the small ones that just not run into the mesh and drop down, they would fly by instinct towards sunlight. And that's why you have a white mesh on top that it intensifies the signal. So the fly flies even higher, and then in that bottle, there's only one exit, and that's this bottle. And that's how we got to these insect soups in the beginning. Each one of those containers represents the bottle content of a 1-week catch. The nice thing about these traps is you can leave them alone for a week, or if you have a bigger bottle, even for longer, and come back, just change the bottle for another one. Many other traps, you have to do it. Imagine the good old sweep net where you have to run around and try to catch any kind of insects. So it's extremely efficient. It catches a lot of specimens, you saw that, and a lot of diversity. But not all of it. I'll pick that as an example, because you can imagine larger insects probably are not fooled by that. Things that crawl on the floor will not make their way up, although we find them sometimes. Sometimes we even find spiders, likely because they ran after their prey and landed in the same trap. And the best thing I've ever seen in a school trap, and I'll show you that, was 2 praying mantises that were waiting next to the exit into the bottle, snapping whatever fly came along. So they were pretty smart now. So we placed these traps all over Canada to make a proof of concept. So for 2-3 years, we went to every single national park here in the nation and put these traps out there, used other traps as well, to catch as much of the diversity of insects. Because that's the part of nature we don't fully understand how diverse it really is. We have a good grasp on the biggies, on mammals, other vertebrates, birds like that. We also involved another helping force, schools. I don't know if anybody who's watching it was part of the so-called School Malaise Trap program. That's why we give out these traps for a 2-week period to a classroom. They can put it in their schoolyards. Probably your schools don't look very diverse to you, or biodiverse, but you'll be surprised. Usually we find at least 60 species in a given time. So even the most concrete wasteland will show that. So we had a lot of these covered across Canada. 6 times we did that program. And then we look at just one curve to give you an idea how diverse just this little fragment of life is in Canada. So we're looking at about 600,000 specimens we went through in national parks, across schools and everything else. We're not done. We found, using our DNA methods, 38,000 species. Known so far in Canada's checklists by experts are about 32,000. So we already have 6,000 more than we knew. And it's a sort of a known fact among experts that we have underestimated diversity in every corner of the planet, but even in Canada, which is sort of uniform, has a lot of ice cover, there's a lot of diversity we don't know. We're not even done. And we haven't even touched things that live in the soil, on the leaflet, and all that kind of stuff. So that the message is here, there's far more diversity out there, but we have technology and collecting methods that use that and utilizes. And the new sequencing methods would actually do that. And one thing, and that's the last one because I'm running out of time, we also ask the public, everyone, to help us with that. We offer this little kit here, it's called Life Scanner, and it works together with an app. So it contains 4 bottles or vials, and you can actually do something Bob sort of indicated. You can take that to the restaurant and take a little sample of your fish fillet or whatever you had, put it in there, take note with your cell phone, send us the data, send us that. We sequence, and you get a message back on your phone, not the same night, unfortunately, but in a couple of days or 2 weeks, you get a message on your phone saying, by the way, that fish fillet was not the right one.